Hello, thanks for attending today's session. The title is Designing IND-enabling Toxicity Studies for Complex Drug Modalities, Going Beyond the Classical Approach. Two speakers, myself, Norbert McCory, and Jennifer Chow. On the agenda today, we'll have some introductory remarks. That will be followed by a discussion of the newer drug modalities, establishment, new established drug and we'll compare with the established drug modalities and the new and advanced modalities. Then we'll discuss optimizing the number of animals on studying the new drug modalities. That will be followed by discussion of regulatory guidelines for these modalities and some examples of juvenile toxicology studies in non-human primates, and also the alternative to the standard mini pig, the nanopig, that is enabling more utility of the pigs as an alternative model. And then finally, we'll discuss the critical milestones in established drug modalities. So just for definition purposes and generalities, we have divided this into generally developmental pathway for well-established drug modalities. And then we have therapies with the evolving regulatory landscape. That's where we have the newer and complex drug modalities. So for this well-established modalities, we have small molecules which have been there for a long time. And in this case here, it's very important to understand what the target identification, the preclinical testing regimen, and then there is manufacturing that comes with that, and finally clinical trials. The second more common drug or established drug modality is monoclonal antibodies. In the early 2000s, these were really the new drug modalities, but because of the number of these drugs developed, approved for use, they have really now become well established. And in this particular case, the therapeutic target is identified, the monoclonal antibody is generated, and then we have the safety efficacy testing, and again, manufacturing and clinical trials. The third is vaccines. Again, vaccines have been here for a long time, right? And even though they could also, some of the vaccines could actually potentially fall into the complex drug modalities because of the application of LNPs and other carriers. They are generally still regarded in the well-established column. So this is by identifying the antigens for the vaccine, interrogate potentials and safety, immunogenicity, and efficacy. And then again, manufacturing and clinical trials. So really well established. Now, when we come to therapies with an evolving regulatory landscape, there are several, right? We have oligonucleotide-based therapies. For example, DNA or RNA, which target nucleic acids, single-stranded SOs, double-stranded RNAs, and saRNA. We also have aptamers, which bind to specific target molecules, and mRNA, which encodes proteins. The second category is gene therapies, right? This is again fairly new. Well, gene therapies has been there for quite some years, but in the recent past, I could comfortably say in the last 2 to 3 years, this has really picked up. We have currently two main delivery modalities, AAV-based or LNP-based. There are other modalities, but these are the main ones that I've mentioned for the sake of this conversation. The third is cell therapy, where we have autologous cells and allogeneic cells. Again, fairly new, but also an advancing field that needs attention in regard to regulatory guidelines and also scientific input. And then finally, I have a category here I call advanced biologics, where we have bispecific antibodies and antibody drug conjugates. Now, antibody drug conjugates have been here a little bit longer, but still, because of the challenges that come with development of this modality, I've deliberately put it in the new and evolving regulatory landscape because there's still a lot to learn, and in regard to making sure that there are appropriate guidelines and regulations for this particular drug modality. Here's a comparison of approvals of this, generally the new versus the more advanced. You will see there that for small molecules, for example, in 2022, there were about 22 approvals. When it comes to monoclonal antibodies, there were 12, ADCs 6, gene therapy, cell therapy about 2. And then as you go to 2023 to 2024, for small molecules and monoclonal antibodies, just staying fairly consistent. ADC seems to have come down a little bit, but it's very clear that this gene therapy, cell therapy, modalities are, even though the numbers are low, but they are increasing in terms of approval. And our estimate is that in the next, 5 years or more, there will really be an uptick once a lot of things have been figured out in regard to this, delivery methodologies and the like. This is another comparison where we looked at the global approvals of the newer drug modalities as of year 2024. And as you can see here, of course, again, monoclonal antibodies, highest number, but gene therapy, again, is right there, 9%, RNAs, 10%, and cell therapy is about 21%. In the classical approach, for example, for small molecule, there are all these, studies that need to be done, right? If you look at the regulations, the guidelines, from efficacy, genetic toxicology, general toxicology, PK, safety pharmacology, ADME-DAT, and carcinogenicity, right? So moving, as we progress on in this conversation, I will focus on general toxicology because a lot of these other types of studies may not necessarily be applicable to the newer drug modalities, and the understanding will become clear as we move on beyond the classical approach in optimizing the number of studies on toxicology studies. So again, I want to give very specific examples where we have seen a dramatic change in terms of study designs, right? Where, unlike the classical, where there was a fixed number of animals and, and that was taken for granted, this has changed. So for example, in small animals, that is in rodents, mostly rats and mice, we now can we have successfully used microsampling, where basically this substitutes the needle and syringe approach, right? And the microsampling actually allows the use of one set of animals. So instead of having a terminal group and TK group, there is no need of that. You can have one group because you are taking very minimal blood from the animals, and so you can do serial collections and also utilize the same animals for monitoring all parameters for your toxicology. Therefore, by the consolidation of satellites, TK animals, and main study groups, in this particular case, there is a dramatic reduction in the number of animals that are needed for study. Secondly, it's very easy to integrate findings, right? Because now you can correlate. For example, if you have taken from the same animals for clinical pathology, it's very easy to relate that to, for example, clinical findings or pathology findings or any body weights and things like that. And finally, this prevents the hematocrit effect associated with dried blood spot analysis. And then another method is the volumetric absorptive microsampling. This is a very specific example. This gives an accurate volume of blood absorbed into the hydrophilic polymeric tip, right? So it simplifies the collection of these the samples. The advantage, again, is the reduction in the number of animals used, right? So for example, in a TK rat study, only 3 animals per dose level for serial collection per animal needed, basically literally reducing the number of animals by almost 50%. In a TK mouse study, again, these are practical real-life examples. In a TK mouse study, for example, only 8 animals per dose level, which are split into 2 subsets. This again reduces the number of animals by almost 60%. So this is just showing that microsampling kit and how it is implemented. So for sampling tips allow sample collections of about 10 to 50 microliters. Then we have a holding case for the tips, which for storage and shipment. This is very fast and convenient packaging, no drying time needed. Other advantages include fast, simple sampling, and no processing of the samples. Again, this just is very efficient and it really cuts down on labor needed. So here are a few case studies for comparing microsampling to the traditional needle and syringe collections. So you will see from this table here that study design, 102 mice could be needed. However, in the microsampling using the Mitra tips, this was reduced to 56 mice, and this study went on successfully. The only downside here is that one must be able to develop a new method for the dry blood collection technique if the previous method was developed in large volumes of blood. In this example, for example, the standard TK rat study would be 90 animals. However, with microsampling, with serial TK samples from the same toxicology cohort, the number of animals again was reduced dramatically. Now we'll switch on to giving examples for large animals. That could be mini pigs or pigs, dogs, or non-human primate, right? So this is just an example of a definitive study in non-human primates. Again, unlike classical study designs, the control animals were reduced to 2 animals per sex. And here we can assess numerous parameters from biodistribution to germline integration to standard assessments. Now, this example here is from a gene therapy study, right? And a single dose, animals are monitored for 3 to 6 months. No need for recovery in this particular case. So in overall, the number of animals was reduced by 10 or more, and that is quite substantial for non-human primate studies. Another approach was to utilize the same animals that were on a toxicology study for a PK ocular assessment, right? So in this case, the toxicology study was designed for using three dose groups plus a control. 24 hours prior to necropsy, these animals were then dosed into the eye, which was collected 24 hours later and assessed for potential biodistribution. Again, this is a gene therapy. Lo and behold, this has proved to be a very successful approach in utilizing the same animals for the parental route of administration for toxicology, but at the same time monitoring for PK for the eye and subsequently selecting a candidate for ocular studies then that can involve imaging and other techniques for ocular toxicology. So again, a very successful approach that can be applied especially for gene therapy and other more complex modalities. Another approach is we really need, EPP&D studies, for example. Probably not with these new drug modalities, because you can use older animals, right? And in one study, you can include fertility endpoints, males and females, but also then perform germline transmission, potential for germline transmission, which you want to be a negative. Now, the animals must be older. And with this approach, you are able to assess microscopic evaluations on any potential reproductive organ, toxicity on reproductive organs, but also take these samples during in life, for example, in males, to look at if there is any germline integration And for females, you can actually collect the oocytes at the end of the study and see whether there is potential for germline integration in the oocytes. In this case, depending on the results, it has become fairly standard now to actually argue against performing a standalone EBP and this study in NHPs, which in any case have become very rare. So there are several guidelines that have been generated in the past few years relating to these new drug modalities. So if you look at the extreme left, we have the FDA and then there's the ICH, right? So for example, if you look at starting, the latest in 2024, there have been guidelines issued by the FDA for ASOs and gene therapy. So the first one is the non-clinical safety assessment of oligonucleotide-based therapies. Remember, even though these therapies have been there for some time, it was all based on case-by-case basis. But now, based on experience, the agency has been able to come up with a fairly, detailed guideline on how to assess the ASOs for in clinical studies. And then we have frequently asked questions developing cellular and gene therapy products, released just November of 2024. And then earlier in the year in 2024, we had two guidelines issued: human gene therapy products incorporating human genome editing, and considerations for the development of chimeric antigen receptor CAR T cell products. And then in 2022, we had a guideline issued on human gene therapy for neurodegenerative diseases. In 2020, there were three guidelines, right? Human gene therapy for hemophilia, human gene therapies for rare diseases, and human gene therapies for retinal diseases. And then in 2013, we have preclinical assessment of investigational cellular and gene therapy products. The ICH also had guidelines, non-clinical biodistribution consideration for gene therapy products in 2023, and 2006 issued general principles for investigating and addressing risks for inadvertent germline integration. Now, the common theme of these guidelines can be generally be divided into two tiers, right? Tier 1 and Tier 2. Tier 1 here, this is the way we look at this, is that this adopts existing regulatory structures, right? And incorporates additional details. For example, in 2013, preclinical assessment of investigation of cellular and gene therapy products was fully adopted in the following, right? I mentioned 2020 human gene therapy for hemophilia, human gene therapy for rare diseases, human gene therapy for retinal diseases. All these refer back to if you want to conduct your detailed preclinical assessment, it referred back to the 2013 guideline. And then the Tier 2 is when it's not possible to use existing regulatory structures, thereby really requiring creation of new frameworks. For example, in 2024, the non-clinical safety assessment of oligonucleotide-based therapeutics is very detailed and includes all aspects of preclinical testing. That is the species, the rationale for species selection, number of animals, and all that. So really interesting way of how these guidelines are evolving in regard to the new drug modalities. So I'll go on to discuss a little bit more specifically some of these guidelines: preclinical assessment of investigation of cellular and gene therapy products, right? Here the animal species is selected based really on the response, biological response, which must be similar to that in human. Some factors to be considered include physiology, anatomy, permissiveness to infection and replication of the vectors, because these are, for gene therapy, either viral or microbial, immune tolerance similar to human, and feasibility of using clinical delivery system or procedures. Now, there is also a description of the use of animal models of disease or injury and proof of concept studies. In addition, animal species in which the gene cell or gene therapy product is biologically active should be used in the toxicology studies. Really important. Considerations for toxicology study designs include clinical indication, anticipated toxicities or adverse effects, any existing pharmacology or proof of concept data, previous experience with clinical delivery device delivery procedure, biological responsiveness of the animal species, putative mode of action, intrinsic properties and pathophysiology of the animal models of choice. In addition, considerations for toxicology design include an adequate number of animals per sex, appropriate control groups, multiple dose groups that bracket the proposed clinical dose levels that bracket the proposed dose range, a dosing schedule that reflects the intended clinical dosing regimen. Multiple sacrifice time points are necessary in order to capture potential acute, chronic, and or delayed onsets of toxicities. And then finally, potential for resolution of any toxicities that have been observed. And then the 2024 non-clinical safety assessment of oligonucleotide-based therapies has also some significant points I wanted to highlight. So this includes toxicity studies conducted in rodents and non-rodents. One of these species should be or must be pharmacologically relevant. The design of toxicity studies is similar to those of the drug of interest, right? Duration of chronic toxicity studies should be consistent with the recommendations of ICH-M3 revision 2. Dosing frequency and duration of the study should consider aspects of the drug, right? Including distribution, half-life, can be shorter in animals than in humans, that's perfectly okay. Dosing in animals generally is more frequent when compared to humans, but also the clinical dosing regimen has to be kept in mind as one designs these studies. Examples of essential study items not covered in some of these regulations include, for example, immune suppression regimens, right? We have seen in the more recent and advanced therapeutics, for example, AAV-based or LNP-based, that immune suppression is essential to tamper down the potential for the innate immune response, for example, excessive cytokines or complement that could result in unanticipated clinical events, preventing the assessment of toxicity. So AAVs, LNPs, and other delivery modalities. Juvenile toxicology in NHPs is also another aspect that is not fully spelled out in these guidelines. And I think in the future, we anticipate to see guidelines coming out to address all these items from immune response to juvenile toxicology. So just to summarize, the unique procedures, new drug modalities include gene and cell therapy and ESOs, CNS dosing, various injection techniques, for example, intrathecal, intracerebroventricular, intracisternoid magna. Again, these are become more commonplace because of the requirement, for example, to inject directly into the CNS. In addition, there is more specialized tissue collections, for example, during in-life, right? This was not common several years ago, but now it's very regular to collect, for example, liver biopsy or liver wedge, a large, from 100 milligrams to 150 during as a major surgery. And then we have biopsies of various tissues, the liver, the skin, lymph nodes, muscle, and lymph nodes. Again, these are collected during in-life to enable correct monitoring of the activity of the particular, new drug modality of interest. Then we have the PCR of these tissues as a molecular biology aspects. So these are tissues for biodistribution, but we also have, as I mentioned earlier on, oocytes from the female animals and semen from the male animals, looking at any potential for integration. So I'll now switch on and just give a few examples of some of the items that have not been addressed. For example, juvenile non-human primates. The goal of this particular example is to show that actually sometimes there may be requirements to dose very early on in juvenile in humans, But what are the appropriate age animals that one needs to use? Now, juvenile animals, or especially non-human primates, can pose several challenges, right? This may require, for example, animals that are 12 months or younger, which creates problems in terms of transferring animals. Breeding to those on site is not a very good option. But also these animals, because they are smaller, sometimes 800 grams to 1 kilogram, lower blood volumes, right? And if the technicians are not adequately trained, it can be a challenge in terms of blood sample collection and even venous catheter placement due to the small veins. When you look, for example, this is an example from our breeding colony. You can see here, for example, from 2020 to 2024, you do not, as expected, get 50 to 50% ratio between males and females. And therefore, again, this can be a challenge when you are trying to dose animals that are less than 12 months old. However, the moment you go beyond 12 months of age, it becomes relatively easy then to get the gender ratio, because then you have a larger population of animals to select from. In terms of immunological response, we have demonstrated and shown that similar responses to girl age in males and females is standard, right? You really do not see any major differences. And in terms of IgG and IgM values, which changed appropriately as expected, we do not actually see any difference, or hardly saw any difference, between animals that were 90 days or all the way to 270. What does that mean? Because we do not see these differences in immunological responses, and we did see similar trends in flow cytometry and TDA values, in addition to minor background microscopic findings, it's really, one can argue that you really do not need to go that young in order to dose, to have a proper animal model in terms of juvenile for your program. You can go animals that are slightly older, more than 12 months, maybe up to 15 months, and still get same or similar results, because again, the major organ systems, including the immune system or the CNS, is very well developed at that time. Finally, I wanted to mention that now, with the efforts to reduce the use of dog and or NHPs, there is a need to develop the mini pig model, right? We know the mini pigs can grow to be relatively large. However, for example, Altasciences, we have made efforts to, over the past 12 to 14 years, to use selection to breed down to a smaller mini pig, which we have called the Nanopig. And therefore, as you can see from these charts here, this has enabled us actually to get an animal that is fairly, weighs almost the same weight from, up to 10 months of age relative to the beagle dog. And which means, therefore, that the same amount of test article can actually be utilized, meaning that we can conduct more studies now using the parenteral route in these nanopigs. So these are some of the important and essential efforts being made to reduce, again, using either the dog and or non-human primates by switching to the nanopig. So as I said, body weight similar to the dog up to 10 to 14 months. We have seen restoration in clinical pathology, low incidence of background pathology findings, and differences in toxicology relevant data are minor in magnitude and biological significance. For example, clinical pathology, organ weights, and background pathology findings. Again, this is really going to ensure that we continue toxicology testing in an appropriate species, but also in terms of test article, use the same material we could basically be using in the dog. So now I'll hand over to the next speaker, Jen, who will take you through some of the case studies and packages that we have seen in the traditional versus new drug modalities.
All right, thank you, Norbert, for that introduction. As Norbert mentioned, my name is Jennifer Chown, and I am the Director of Strategic Programs here at Altasciences. And I'm happy to be joining you in this webinar today, particularly to cover how your contract research organization's program management team plays a vital role in ensuring that your IND studies are executed with precision, speed, and strategic oversight, most especially for therapies of evolving regulatory landscape. At Altasciences, the program management team will work closely with business development and scientific experts to develop tailored Gantt charts for your particular program. This showing here is an example of a program overview for a large molecule with well-established development pathways, such as a monoclonal antibody, again, as Norbert had listed at the beginning of his presentation. This program depiction projects the feasibility of achieving CTA submission in 34 weeks and IND submission in 40 weeks from test article availability. It is very important to note, however, that for more complex emerging large molecules, such as gene therapies, cell-based therapies, and advanced biologics, the drug development pathways will be much more variable and dependent on the specific compound characteristics and target. Additional time needs to be factored in for any such emerging compounds, such as ADCs, for instance, where bioanalytical assays will be required for the total antibody, the total conjugated payload, and the free payload, as well as an ex vivo stability study required to assess how stable the ADC is in the biological matrix outside of the organism under controlled physiological-like conditions. Similarly, for gene therapies where PCR is required to assess biodistribution, additional time will be required due to the complexities that go into PCR, tissue collection, processing, and analysis, all of which take longer than the other assay types. Any compounds that require tissue bioanalytical analysis or immunohistochemistry will also require more time, as will any drugs suspected of modulating immune function, which would require a T-cell dependent antibody response assay, again, simply due to the elevated complexity associated with deriving these endpoints. This example of a Gantt chart is of a typical small molecule program run here at Altasciences. There are many advantages to maintaining the bioanalytical within a single CRO, and even more so when co-localized with the in-life, which we'll get into in a bit more detail in some upcoming slides. As a fully integrated CRO here at Altasciences, we offer drug manufacturing support out of our CDMO site in Philadelphia, and our CDMO team works in tandem with the preclinical and clinical teams throughout the entire drug development process. Partnering with Altasciences for all of your drug development needs, CDMO, bioanalytical, preclinical, and clinical helps to ensure open, direct lines of communication between the sponsor and all teams throughout the entire process, all overseen by one dedicated program manager from start to finish. Optimizing this model helps proactively ensure that milestones are met and that timelines are maintained on track, or that any issues are promptly communicated and addressed and mitigated. Here at Altasciences, our regulatory and scientific affairs teams can also help support all of your submission needs for two regulatory pathways, CTA and IND, again providing opportunity to save in your overall drug development process. So how do program managers help contribute to a successful IND program? At Altasciences, our program managers work closely with scientific and regulatory leads to ensure that your program design is informed by regulatory expectations and scientific feasibility. Where applicable, we help align non-clinical and CMC strategies to support clinical trial readiness. A dedicated program manager is assigned at contract signature. This provides a centralized point of contact for the client and internal teams, enabling enhanced efficiency and proactive milestone tracking. This also helps identify and address potential bottlenecks early on in the process. The program manager will work with business development and the internal teams to develop a tailored summary Gantt based on therapeutic modality, whether you're working with a small molecule, a biologic, or a gene therapy, for example. The Gantt will include modalities, specific requirements, and risk areas, ensuring that expectations and feasibility are aligned from the start. The program manager coordinates handoffs from non-clinical to clinical with minimal delays and aligns bioanalytical methods, formulation, and regulatory documentation to support a seamless crossover, reducing downtime and duplication. Program managers are not simply responsible to track, but are responsible to support sponsors as strategic partners. They can connect people, data, and decisions across functions and enable IND success through rigor, customization, and collaboration. The program and project management teams at Altasciences use a series of integrated tools to enable communication and integration across services and sites, which ensures the seamless client experience. Our proprietary database behind our commitment to communication, Ask Albert, allows for timely information capture and proactive communication across all teams, departments, and sites for smooth transitions between phases and studies. When working with any of Altasciences' service lines, you are supported by program managers for non-clinical IND packages or CDMO packages, and by clinical project managers for your clinical studies. In addition, for cross-functional programs, you will be supported by a cross-functional program manager who will be a consistent point of contact, helping you navigate the complexities of your drug development pathway and reinforces expectations at every step of the process. Across all areas, our program and project managers follow consistent key startup tasks with formal kickoff meetings followed by regularly scheduled meetings to maintain open communication. Through utilization of our proprietary centralized scheduling system, everyone has shared visibility to completed milestones or scheduling delays to ensure that everyone is working in unison and timeline delays are promptly mitigated. Customized summary reports are provided to our clients, as well as tailored dashboards to help provide consistent, constant communication and insight for our clients to utilize to maintain their wider teams informed. The program and project managers are critical also in assessing potential risk, mitigating risk, and resolving issues. Particularly for therapies with evolving regulatory landscape and complex cross-functional integration, proactive planning can help protect timelines, quality, and outcomes. Complex programs rely on tight coordination across multiple functions. A failure or delay in one area can create a domino effect, impacting the entire timeline. Clear awareness and mapping of all interdependencies is absolutely critical. The newer or less established modalities bring greater uncertainty and increased risk of unanticipated issues. It is critical to avoid cutting timelines. Unrealistic timelines increase pressure and reduce capacity for proper issue resolution. Program managers must advocate for reality. It's essential that program managers should push back on aggressive assumptions that don't reflect the scientific maturity of the modality, and build in buffer time and contingency planning, especially for first-in-human or novel trial designs. It's also important to use data from similar programs to inform realistic estimates. Balancing innovation with discipline, speed is important, but predictability and quality are critical. A well-paced plan reduces risk, enhances quality, and supports regulatory confidence. Program managers act as the central hub across functions, prioritize close monitoring of high-risk, high-impact milestones, and proactively flag and escalate potential delays or gaps. As you have seen throughout this presentation, as a fully integrated CRO, Altasciences has the necessary services and scientific expertise to help you accomplish your drug development goals. Program and project management work across the entire end-to-end solution, whether you're placing a single study with a single site, multiple studies in a particular site or service line, or engaging us to partner with you on the full drug development pathway of a particular compound from discovery through clinical, with regulatory support, into first-in-human clinical, with bioanalytical support optimally integrated throughout all the process with a single CRO provider. Program managers are here to help be an advocate and a voice for you within Altasciences through the entirety of your drug development journey. Leveraging the lab as the bridge across your journey is the optimal approach for reduction in timelines and costs. Co-locating bioanalytical labs with in-life facilities streamlines logistics and enables real-time communication, as well as faster decision-making while reducing sample transport time and complexity. It equates to accelerated method development, leveraging preclinical bioanalytical experience for human method development, anticipates timing and regulatory needs for clinical transition, reduces costs by avoiding redundant development work, and eliminates scheduling bottlenecks in clinical workflows. Timed sample analysis helps drive data and decision-making aligned with clinical study needs. And rapid data turnaround supports real-time decision-making and ensures studies progress based on data and not delays. The lab stays involved through all of the development phases, ensuring analytical consistency from preclinical through to clinical, and builds cumulative knowledge of the molecule for optimized support. Internal data flow equals faster reporting. Centralized data handling accelerates reporting cycles and facilitates quick updates to regulatory documents, INDs or CTAs, as well as enhanced data integrity and audit readiness. A single cross-functional program manager oversees the full workflow, aligns preclinical, clinical, and analytical timelines, and ensures continuity, accountability, and strategic oversight. In summary, Altasciences provides an end-to-end solution with oversight by one dedicated cross-functional program manager, which reduces clients' management and vendor burden and ensures robust tracking, communication, risk mitigation, review of emerging data, and pivoting in real time to program challenges or opportunities. All right. Any questions? Okay, so I see that we do have some questions in the chat. The first question is, what additional data in NHP infants shows similarity in development? I think Norbert, if you can answer that.
Yes, that's a very good question. So in addition to immunological data, we have demonstrated using, learning and memory via the Wisconsin General testing apparatus that actually even neurologically, the developmental, paradigm is very similar between infants aged 9 months all the way to 14 months. What that means is that, again, we really do not need to, we can test infants that are greater than 12 months of age and satisfy the scientific and or regulatory needs without reverting to testing infants that are young and very difficult to obtain.
All right, thanks, Norbert. There is another question. It's, how do you assure contaminant-free oocytes are collected in NHPs for PCR?
Another good question. So this can be very tricky, right? In terms of assuring that the material you are collecting has no contaminant that could potentially result in false negatives, right? So this is a very intense process, and absolutely proper training and proper techniques are required that include, after collection, basically carefully removing all any adherents or any cells around the oocytes. And this has been, the techniques have been developed in conjunction with a well-established laboratory in an academia where they have been doing this work for years. So we have shown and are confident that the oocytes that we collect using this technique are of utility. And finally, we have to make sure that our technical staff are continually trained using oocytes from other species with the goal of assuring that they are able to collect these samples with no contamination at all.
All right, perfect. I do see another question here. It's, what is the optimal method for processing NHP semen for PCR?
So that's another very good question. So basically, there are two potential methods, right? There is direct cryopreservation, where the semen samples are collected, mixed with equal parts of preservation buffer and stored. Or there's what we call the swim-up method, where semen is initially incubated with sperm preparation medium, which draws the motile sperm to the top layer. That sample is then collected, again, cryopreserved and stored for subsequent utility. We have found that both methods actually give fairly similar amount of DNA, RNA when analyzed for PCR. All right, we see another final question here. In your experience, have you seen a standard approach to toxicology study designs for gene therapy?
Yeah, so so far there are no standard designs as of yet. Each product is unique and really requires careful review before designing study types and the study types that are needed.
So those are all the questions that we have seen currently. Thanks so much for attending this session. Please feel free to send any additional questions and we'll be more than happy to respond to you. Again, thank you very much for attending the session.
Thank you.